Here we see about glycolipids. It is also one of the compound lipid. It is the conjugated lipid because the carbohydrate as a prosthetic group combined with lipids, so that produce glycolipids. These are the substances that is the combined mixture of lipid and carbohydrate at certain proportion. The glycolipids are important constituent of cell membrane and a nervous tissue, particularly the brain. They contain a ceramide equal to sphingosine and fatty acid. Sphingosine attached with the fatty acid, we called it as ceramide. And also one or more sugar units combined in here. So that is glycolipids. There are two types, cerebrosides and gangliosides. First, we see about cerebrosides. It is also known as glycospingosides. It is occur in large amount in the white matter of brain and in the myelin sheath of nerve. So these structures made up of the cerebrosides. They are not found in embryonic brain but develops as medullation progresses. So later stage only that is formed inside of brain. In smaller amounts, they appear to be very widely distributed in animal tissues. In medullated nerves, the concentration of cerebrosides are much higher than in non-medullated nerve fibers. Structure, it contains sugar, usually galactose, sometimes the glucose. And the high molecular weight fatty acid is present and also alcohol. The sphingosine is the alcohol. When we hydrolysis of any one of the cerebroside, we get following components also. These are derived lipids. Next, types of cerebrosides. The individual cerebrosides are differentiated by kinds of fatty acids in the molecule, which means which fatty acid present in here based on this that are differentiated. First one, carrasine. Carrasine contain normal lignoceric acid, that's the molecular structure is C23H47COOH as fatty acid, and lignoceric acid is synthesized from acetate by repeated condensation of C2 units, which means the acetate undergoes repeated condensation of a C2 units, so we get lignoceric acid. It is the fatty acid. Next is cerebron, also prionosine. It contains hydroxylignoceric acid, also called cerebronic acid, which means the hydroxylation process occur instead of lignoceric acid and add a hydroxyl group, so we get cerebron. Next one, nervon. It contains an unsaturated homolog of lignoceric acid called nervonic acid. Nervonic acid has a double bond appear to be formed by elongation of oleic acid. So from C18 to C24 monosaturated fatty acid. In these regions, the changes occur, so we get the nervon. Then oxynervon contain hydroxy derivative of nervonic acid. So there are four types of cerebrosides present in our brain: carrasine, cerebron, nervon, oxynervon. Next we say about psychosin. The prolonged hydrolysis of any cerebroside with barium hydroxide, fatty acid is removed and it yields psychosin. Psychosin can be further hydrolyzed to yield sphingosine and galactose. Then clinical aspects. The Gaucher disease is caused by the disorder of cerebroside metabolism, also known as lipidosis. It is the inheritable disease and it is considered as the autosomal recessive disorder, and enzyme defect due to the enzyme beta glucocerebrosides. It is the lysosomal enzyme. So due to this enzyme defect, the disease is occurred. In absence of this enzyme, the cerebrosides cannot be degraded in the body. So result is glucocerebrosides, usually carrasine, accumulate in reticuloendothelial cells, while the liver, spleen, bone marrow, and are also brain. So complex lipids appear to collect within mitochondria of the reticuloendothelial cell. So the elevated serum acid phosphatase ACP level is increased. So the clinical feature of that disease, adult as well as infants are affected, and in infancy and childhood, fairly acute onset with rapid course and death in several years. The infant loses weight, fail to grow, progressive mental retardation. There is no growth for the children's. Initially, there is spasticity, later on followed by flaccidity. So next, in adult, the condition is progressive enlargement of spleen. Splenomegaly is occur. Splenomegaly means enlargement of spleen, which may reach to umbilicus or below. Characteristic of bone pain due to marrow cells replaced by histocytes loaded with lipids. As a result, totally, the simply called as the lipid storage. As a result, lead to progressive anemia, leukopenia, and thrombocytopenia, tendency to get secondary infections and a bleeding tendency. Next type we see about for the glycophospholipids, gangliosides. It is the highest concentration are found in gray matter of brain. Gangliosides are most complex of the glycosphingolipids. They are large complex lipids. Their molecular weight varies from 1,80,000 to 2,50,000. And structure, we see the structure. It contain a long chain fatty acid, usually C18 to C24, then alcohol sphingosine, a carbohydrate moiety, usually glucose or galactose. In sometimes one molecule of N-acetyl galactosamine is present. At least one molecule of N-acetyl neuraminic acid, also called sialic acid, is present here. Then types of gangliosides. Over 30 types of gangliosides have been isolated from brain tissue. Four importance are major: GM1, GM2, GM1, GM2, GM3, and GD3. Clinical importance: the simplest and common ganglioside found in tissues is GM3, which contains ceramide, one molecule of glucose, one molecule of galactose, and one molecule of neuraminic acid. Then also GM1 is the complex ganglioside. It is considerable biological interest due to it is known as the receptor in human intestine for cholera so it can accept the cholera. Next, functions of glycolipids. These are the important constituents of nervous tissue, such as brain and outer leaflet of all cell membrane. They play a role in regulation of cellular interactions, growth and development. Glycolipids serve as cell surface receptor for certain hormones and a number of drugs. They also serve as receptor for cholera and tetanus toxin. So next, the glycolipids are antigenic and they have been identified as a source of blood group antigens.